Okay, we will proceed with our distinguished lecture, Dr. Marco Andreani. Today we have the honor to be with us the president of the European Federation for Immunogenetics, Dr. Marco Andreani. On behalf of the Hellenic Society of Immunology, I would like to thank him for accepting our invitation to participate as invited speaker in the 13th Pan-Hellenic Congress of Immunology. Dr. Andreani is a distinguished scientist in the field of histocompatibility and transplantation immunology, involved in both hemopoietic stem cell and solid organ transplantation, as well as in population genetics and immunological tolerance. Dr. Andreani is now the director of the Transplant Immunogenetics Laboratory at the famous pediatric hospital Bambino Gesù in Rome, Italy, where he is also a member of the ethic committee. So it's author or co-author in more than 160 publications in peer-reviewed scientific journals. But beside his scientific activities, I would like to focus in his involvement in European Federation of Immunogenetics, EFI. He is the president of the European Federation, and he was also Chancellor of the EFI Executive Committee from 2019 to 2022, member of the EFI Education Committee for 3 terms, past chair of the European Board of Transplant Immunology, member of the EFI Population Genetics Working Group, and of course, EFI Inspector and EFI Commissioner. So, I know Marco since several years now, but I had the opportunity to know him better, to collaborate and appreciate his dedication to EFI during our joint participation in EFI Executive Committee in both of his terms. And now I would like to ask from my co-chair, Dr. Chryssia Papasteriadis, to welcome also Marco.
Thank you, Katerina. Okay. Thank you, Katerina. Before welcoming our distinguished speaker, Dr. Marco Andreani, and with your permission, dear Marco, I would like to say that it's a pleasure to me to co-chair this session with Katerina. Unfortunately, Yannis Baltadakis has some personal problem. He couldn't be with us. So I have to say that Katerina used to be a medical resident of mine in the Evangelismos Hospital, and for a long time co-worker at the same hospital. So, dear participants and dear colleagues, it's really my special privilege and great joy for me to also welcome to our conference Dr. Marco Andreani. I had the opportunity of working with him for many years within EFI's several activities. His dedication, marked by diligence, effectiveness, humility, and commitment to the scientific committee, has been evident throughout his service, both in his home country and in various roles he has held and continues to hold across Europe in the field of immunology, immunogenetics, and transplantation. Despite his many responsibilities, he has contributed to advanced science, so today he leads the very demanding transplant immunogenetics lab, as we heard before, at the Vatican's famous hospital. And of course, it's no coincidence that today he serves as EFI president. HLA matching and cure in hematopoietic stem cell transplantation is the title of his lecture today. An interesting topic, since treatment of patients with several and certain high-risk hematological diseases and beyond is only possible with hematopoietic stem cell transplantation. Selecting the best donor is a complex process. The key criterion in this procedure is compatibility in HLA antigens between donor and recipient. A fully compatible donor is the best choice. And if this is not the case, in the so-called alternative cases, cell donor is selected taking into consideration recipients, class 1 typing, HLA class 1 typing, and donors KIR genotyping, aiming to optimize graft versus leukemia activity while minimizing graft versus host disease, improving at this way patients' survival. Now, I am really glad to give the floor to Dr. Andriani, to my good friend Marco, to guide us through the paths of hematopoietic stem transplantation and to share with us his really great experience in this field. Thank you.
Well, thank you so much for this introduction, and I promise that sometimes I feel down a little bit. I will come here to talk to you because it's such a big pleasure, and thank you for the invitation, Katarina and Krista, for our friendship and all for the work we have done together. So today I will talk about, as Krista was introducing, HLA matching and KIR in stem cell transplantation. It's a difficult task because there are so many variabilities that are involved in choosing a donor, and there are also many complex potential risk factor and variabilities, including the stem cell sources, other factors like the conditioning regimens, the disease progression, and of course there are some issues like CMV infection of the donor and of the recipient, the donor age, that is very important, and donor gender. But as we all know, also HLA, and that's why I'm here. I'm sorry, I'm not an immunologist. I am an immunogenetics, like maybe probably you know, and I hope that you will find some sort of conversion in the two fields, because I don't need to tell to you what HLA is, but just to remind that it's the most polymorphic system in our body, and it's located in chromosome 6. It's divided in the genes are from class 1 called A, B, C, and class 2, the RB1, DQ, and DP, that are the classical one. There are an expression of molecules on the surface of the cells and in a covalent way. And as you all know, these are the pocket where the pathogens or the immune peptidome that we will see a little bit more in details later are sitting in order to be presented to the T cells and to the immune system in order to have a reaction. So I will tell you a story today about the different many aspects that characterize the continuous work of people like us that are working in the lab and the clinicians that are working in the hospitals. And the major point I will focus on will be little chapter. The first one will be HLA matching versus mismatching in MAD, meaning marrow of unrelated donors, so donors that comes from the registry. And the big story was permissive HLA mismatch, because some, as you know, of the mismatch between donor and recipient are permissive, meaning that they do not offer a possibility of rejection of graft versus host disease. And then I will approach the field of HLA matching in haploidentical transplantation that, as Krista was pointing out, are it is a very important topic in these days. Let's start from what we know. We all know that HLA matching is important. I would like to start from this collaborative work that we did in Italy a few years ago, in which we clearly indicated, as many others group, that to be matched in order to have a successful transplant is much better than having a mismatch in the HLA. This for overall survival, for graft versus host disease, for non-related mortality, and this is very clear. One of the things that was very interesting in that work is that taking out only a group of patients of Italian origin and the donor of Italian origin, the incidence of graft versus host disease was much lower, meaning that probably there is something related to HLA included in the haplotypes. And this is a topic that needs to be discussed and investigated much more. But let's take this answer also in a much bigger group. There is a very recent paper from Esteban Bolanos from Essen, in which he was investigated more than 17,000 patients, in which again the matching is much better than not matching for 9 or 8. And the major point was to have not to have a matching, not to have a mismatch in the class 1 loci. As you know, the introduction of post-transplant cyclophosphamide has been one of the most important improvements in the recent years in terms of helping the survival of a patient. So the question was, does the use of post-transplant PCR really improve the clinical outcome in a matching setting? And there are two different studies, one from EBMT and the other one from CI-BMTR. The first one from EBMT shows that, of course, the use of post-transplant PCR, post-transplant cyclophosphamide, is helpful. So the patient that receive a post-transplant cyclophosphamide perform better than the others. And this also in terms of risk factor for graft versus host disease. And this is true also in the other survey from the CI-BMTR, in which, again, comparing the group of patients treated with post-transplant cyclophosphamide respect to the conventional treatment, perform in a better way. But the question is, does post-transplant cyclophosphamide improve the outcome when we are treating patients with a mismatch, meaning with some differences in the compatibility between the donor and the recipient? Well, there are two studies, again, one from EBMT and the other one from CI-BMTR, and we have different answers. The first studies say yes. If they took out only the patient that received a post-transplant cyclophosphamide, and the differences in terms of mismatching is exactly the same, like in the conventional treatment. But this was not the case in the other study. So they say that if you treat a patient with a post-transplant cyclophosphamide, you don't need to have a full match donor. This is something that for people that is taking care of HLA typing since a long time, besides where, as we will see later on, the introduction of haploidentical, meaning that you receive transplant from a donor that is completely different for one haplotype, is a very tricky argument. Now I will want to go a little bit before to move on the what I call the background, what we know. We have done trying to find a solution to use mismatched donor that were permissive, meaning with permissive that didn't have any impact on the outcome, even if they were different at the HLA level. there is this first study from Cavase since 2007. It's an old study in which he was able to identify some position in the task of a HLA packet in which he was able to define matching versus mismatching and a better outcome. Then there is this other work with Effie Pedersdorf from Seattle in which she was saying that the differences at locus C in the terms of expression between donor and recipient was considered a risk factor for the patient. But we know, for example, from another study from the American group led by Marcelo Vigna, in which he was able to define the permissive combination between HLA-C of 3 or 4 that were different, but didn't influence the outcome of a transplant. That's why we consider this permissive matching. Or another study from the Japan group in which they defined that the differences at two different loci, C1402 or B1501, were detrimental for the transplant in terms of graft versus host disease. Then there is this big news story, quite new, that is the B leader role. We know that in chromosome, in exon 1, in HLA-B locus, there could be a polymorphism between threonine and methionine. And the group investigated more than 17,000 patients, trying to define the differences. There were differences in the outcome, clinical outcome, in terms of differences between the leaders. So they consider all the matching, comparing the patients that were mismatched only for the B locus, and they defined that to have a methionine heterozygous situation with threonine or homozygous was a risk factor for the transplant. And this is something that you need to take care of, also because they also further investigated patients that were completely matched for the B locus, but mismatched for only one of the other loci. And again, it came out that the presence of MM was a detrimental aspect for the transplant. And it's easy to check if you want, who is taking care of the laboratories, to check this information, because there is a free website, IMGT, where you can put the donor typing of a B and the patient typing of a B, and to receive an answer in order to know if this combination of M and T is detrimental or not. And then, of course, talking about permissive mismatch. Again, permissive meaning that we try to define a donor that even if it's different from the recipient, will not affect the clinical outcome. And of course, we cannot not to talk about the DPB1 experience from the group of Katharina Fleischauer, and what we also consider in immune peptidome. There are, as you probably, the people that are taking care of a laboratory like that I see in our laboratory of histocompatibility that is in the room, we know that there are two different models. One is what is called the T-cell epitopes model, TCE model, and the second one is the expression model. The first one has been able to define for only the DPB1 locus the possibility that some combinations are permissive and other are not permissive, defining 3 different groups, group 1, 2, and 3. So having the possibility to make a transplant between donor and recipient all belonging for the DPB1 to group 3, the survival was much better. And this was, in another way, confirmed by the model of expression. Effie Pedersen was able to show that flanking to the genes of HLA, there are two specific genes, two specific polymorphisms that we call A and G, this long way to nominate the gene, and they find out that if in a condition in which a donor was A homozygous, the outcome was much better respect to the other. And now we are combining these two models. So I know that from an immunologic point of view, probably this could be not relevant, but for people that is taking care of a transplantation, it's a very important information. That's why we have learned how to type DPB1 and to take care of these and to give this information to the clinician. But the people that is taking care of a DPB1 locus that has become a model has further investigated the approach. And as I was showing you before, we know that the immune peptidome is a bunch of peptides that are sitting on the pocket of a HLA. And they can be, if they are presented by class 1, they are something like this. There are some anchor where the different types of peptide can be presented to the T lymphocyte or to the immune system. And this is just a little bit different for the class 2, but the concept is exactly the same. So we have a pocket in which the peptide, all the potential peptides are sitting there. And so taking advantage of a model, they were able to investigate the fact that within the different, in the algorithm, between the different groups that they have defined, like more immunogenetic or less immunogenetic, they understood that there are in less immunogenetic, for example, like in a group 3-3, the number of in immune peptidome that was presented was much less. And so this results in a less the presence of a lower number of T clones that could be presented aggressive within the pairs of donors and recipients. And this is very important. They further investigated also within the same group, non-core and core group. But this is something that I don't want to discuss too much, but it's still very important. Then they moved to a different approach for class 1. So we have understood that DPB1 is a model, and we can take advantage of the fact that the DP can be permissive, and the fact that they are permissive now it's clear because there are less immune peptidome that are sitting there, that are more similar. So they approached the same issue, trying to understand if in the class 1 they could have something similar. Again, this is the pocket of a class 1 locus. Could be a locus A, for example, or a molecule outside the surface of a cell. These are two different loci, O201 or O201. And they have different anchor that they can, for this reason, they can accept a different immune peptidome, or they can accept the same peptidome, meaning that they are different, but they are not so different like they could be. And this means, again, that the clones of T cells that comes out from this fighting could be less. And they were able to group based on these characteristics in what we call peptide binding motif, PBM, in 21 groups. And they classified like PBM that as a large number of immune peptidome presented like match, and compared these this patient that received a transplant from a donor with a peptide-binding motif that was classified less immunogenic respect to the others. And as you can see from these curves, it is very clear that also in this case we can define not only patients that are completely matched, but also patients that are mismatched, but that have a sort of permissive action between donor and recipient. And this is very important, it is very new. And by the way, this model has been sort of validated by a different paper that came out very recently from the group of Seattle with Mehta and Effie Pedersdorf, in which you can distinguish between the curves. They consider that the best curve is a HLA match in young patient, and this is always true till now. But the second curves compare patients that are mismatched for the PBM, as I showed you just a second ago, but with adult patient completely matched 10 out of 10. So this means that the classification really works, and this is something that in the laboratory we need to take care and we need to consider in the future. So approaching HLA matching in haploidentical. Haploidentical has become something that allowed every one of us to have a potential donor, every one of us, all the clinicians, a potential donor very soon and available immediately. Because you know that to have an unrelated donor, it takes time, you need to type it, you need a confirmation, the blood, the peripheral, the stem cell must travel from far away. Instead, if in a pediatric laboratory, in a pediatric hospital, like the one in which I work, many mothers or fathers are willing to donate their peripheral blood, stem cell peripheral blood to the baby. And as you can see, was fascinating, and none of us a few years ago could believe that this was possible. What I told you till now is somehow not true. Not true because, as you can see, the HLA compatibility doesn't look any longer a problem in haploidentical transplantation. This is a compare of patients that are matched or not matched. And again, in these three graphs, you can see that two donors different for few or more alleles do not affect the outcome of the transplant. And again, this other work. So many papers now are saying that it is possible to make a haploidentical transplant without risk, graft versus host disease or rejection. Also because it came out the use of post-transplant cyclophosphamide, the same that I was telling you before for the MAD experience. There is some exception. There is a very nice paper in which, again, the group of Fax and Pedersdorf showed that the B leader could be important. So if you do not consider the full haplotype different, but you consider only some aspect of that, for example, the B leader could be important, even in the context of a different haplotype, also for the DRB1 or for the DPB1. So picking up only differences relative to these alleles, or at least this is what is available now in the literature. Our experience in Rome is very nice. We have a very nice curves even if Professor Locatelli, that is the responsible of our department, is using alpha-beta depletion, and so we use gamma-delta. It's a T depletion treatment. And as you can see, the curves of haploidentical in pediatric patients affected by malignancies are really incredibly high in terms of survival. But we wanted to take care and to observe, based on the study of Crivello that I was talking to you before on PBM, we wanted to see if the differences classifying our patient with that tool, the PBM, the peptide binding motif difference, there could be some differentiation. And actually, this is what we are observing, and this is very interesting because, again, this could be a very nice way to take care of the typing of a patient, in particular for haploidentical. But there is another big issue for treating haploidentical transplant, that are the presence of donor-specific antibodies. You know that we can have HLA antibody, but we also can have specific, donor-specific antibodies. And as you probably know, there are some recommendations from the EBMT that suggest to everyone to take a look to the number of DSA, donor-specific antibody, present in the recipient before to make an haploidentical. So if you have a possibility to take care of to check the level of antibodies against a father or a mother or to a brother, you can really choose in an excellent way the best donor. Because looking at these curves, that I will show you in a second, you can see that the presence of DSA is really the detrimental aspect for haploidentical transplantation. This was, you can see the first curve, the highest curve is relative to patients that do not have DSA. Then you can see the other, in which you can see that the patient that didn't have HLA antibodies before the transplant, but that develop de novo DSA, or patient that always have the DSA, the outcome, the clinical outcome of this patient is really very low. This was confirmed by another great scientist, Churea, from the Anderson University, in which he was showing that the number of MFI that measure the level, the quantity of antibodies is really very important, and in particular in combination with the presence of C1q. And this is something that we need to take care. In our experience, for example, we transplanted in our hospital 20 thalassaemic patients with haploidentical, and you can see that 7 had a graft failure respect to the 13. The numbers are very low, but thalassaemic patients do not often receive haploidentical transplantation. And you can see that among the group of the patients that rejected the transplant, 71% were positive for DSA. Now, there is before to move to the key aspects that I know are very interesting, I would like to share some information about a different way to type. To type not only HLA, but applets. When we talk about a molecule of HLA, a cell with some specific molecule of HLA, there are not antibodies against all the molecules. There are antibodies against epitopes. And in particular, in epitopes, there are the applets. So applets are very short sequence of nucleotide. There are this table summarize some of them, and this you can see much better. This is a T144KR that is the yellow part, and this is the applet. That is probably what mostly of the time the antibody recognize. So there has been a lot of people that now, this has been very important for the transplant in solid organ transplantation. There are a lot of evidence in which it's very clear that the mismatch at applets level is very important. Now, there has been many studies in which we have different conclusions. This study says that a higher level of applet mismatch between donor and recipient could be detrimental. It was the same for this study from Zhu, but after a few years, he published a different paper englobing many more patients in which he was saying that, well, it's not so clear what is going on. But definitely it's very important. In solid organ transplantation, the matching at applets level is really relevant. We took care of these. We checked in our hospital, in our patient, and actually we didn't find so many differences between patient and donors that were mismatched for a higher number of applets. But and now we go to the last recommendation and the last part of my talk that is relative to the KIR. And again, EBMT recommendation that NK alloreactivity is something that should be taken into consideration, in particular for haploidentical. We know that NK are the natural killer cells. They are acting in a very satisfactory way to take our immune system clear, and there are they fight against tumors, against infected virus cells, and there are two main groups of receptor, lectin like CD94 and KG2 that I'm not talking about today, and the immunoglobulin superfamily of the KIR. Just to remind you, you know better than me what KIR has, but just to remind that are surface molecules with 2 or 3 domains. They have an intradomain structure. They could be active, inhibitory, or not inhibitory. They are located in chromosome 19, and they are divided in haplotype, in 2 different haplotypes, haplotype A and haplotype B. These 2 different haplotypes are characterized by the presence or not of inhibitory or activating genes. The B has more inhibitory activating gene, and A more inhibitory. And not only these inhibitory or alloreactive are located in the telomeric or in the centromeric part. The important things to remember is that there are specific ligands in the HLA system for the KIR, in particular for this one, the HLA-C1 and HLA-C2, they are different because there are groups of HLA-C loss molecules that are different from one to the other because in the position 80 they have a lectin respect to the asparagine. And so they make different groups. And this makes the specific KIRs being a ligand and a receptor, like this. So if the homeostasis of the body is fine, there are no tumor cells, normal cells, activating and inhibitory cells, as you know, do not kill the cells. But if the HLA molecules are missing or there is an overexpression of them, the KIR are able to kill the cells that they do not like. And in this context, in haploidentical context, is very important. And in the times, over the time, there has been developed many different models, too many, but I would like just to summarize to you because I think it's very important. The first one was the ligand-ligand model from the group of Perugia and Ruggeri that she was remembering a few minutes ago before I started, in which basically you didn't need to type the key, right? You just need to type the HLA of a donor and of a recipient for the C and for the B. And there is, again, in IMGT, there is a very nice software in which you can enter the typing of both of them, of both the donor and recipient, and to understand if there is a sort of alloreactivity that could be useful. Then there is the receptor ligand model, in which you have to type the KIR of the donor and the HLA of the recipient. And if you, in the case you miss the presence of in HLA, the KIR could be, the NK could be alloreactive. And this is just a curve of a group in which you can see that the differences in terms of clinical outcome, considering this missing ligand, could be very, very important. Then there is the educational models, because when we transplant the immune system of a donor, some of the KIR do not know exactly what they are. They were educated for the donor, not for the recipient. So sometimes they get a new education, sometimes they don't. And based on this, again, there is a specific way to follow the outcome of a patient. Then there is what the Cooley's work came out with differentiating the two haplotype. The haplotype that consider more inhibitory or more active genes inside. And again, there is a very nice tool in which inserting the typing of the donor and the recipient, you can receive for that specific model a sort of prognostic aspect of the outcome. And these are curves that comes from these observations. And then there is the matching model that we can skip. But basically, what I would like to share with that, there are so many models. Some are specific for some specific disease, has been observed to be working for specific disease. Some as contradictory results. And what I found very interesting was that in the time of post-transplant cyclophosphamide treatment, there was this nice paper from the group of Churea in which they were able to differentiate, to count what they call the count functional inhibitory KIR score. So they made a KIR based on the number of inhibitory KIR, and they compared all the different models that I presented to you today. And actually, they were able to demonstrate that this new way in patient transplanted for malignant hematological malignancies in a post-transplant cyclophosphamide environment acted really very, very much better than the other. And the second work from a Spanish group was basically confirming these results. Well, the ABMT is coming out with new recommendation. I have the honor to be in the group of these people. So soon there will be new guidelines that probably will take care also of this. There will be also the story of HLA loss that people taking care of a laboratory know what I'm talking about, but we don't know. We are in the problem in the moment in which there are so many information, so many differences, so many variabilities in which we probably, even if I don't like to say that, we probably will need specific artificial intelligence to put everything together, and new machine learning to try to find an help in a sort of consulting. This is too early to say, but probably this is something that in the future we will need to take care of. And so just to give you a final message, the HLA compatibility is still better 10 out of 10 than 9 out of 10 or 8 out of 10. This is clear in mud environment. But the use of post-transplant cyclophosphamide increases the probability of a higher overall survival, also in MAD, in marrow of unrelated donor, not only in haploidentical. Still debating the importance of LA matching after the use of post-transplant cyclophosphamide. We don't know if among the two surveys that I show who is really right. So should we consider the HLA typing not longer important for if we treat the patient with post-transplant cyclophosphamide? We need to understand that there are environments in which permissive mismatch are important and should be considered. The presence of antibodies against the donor, the DSA, the donor-specific antibodies, are very important, in particular in haploidentical transplant, but also in MAD, if we know that the specific antibodies are against the locus that is different. The role of applet matching, so the way in which we can type at a different level, the typing between donor and recipient should be investigated much more, as well as the role of a NK of a KIR. And so the use of artificial intelligence, maybe that the next time that we will meet, we will have something to say, I don't know. Just to thank to all the people that allowed me to be here, close to many friends are working in Rome and in Essen, like Caterina Fleschauer, Crivello, that we are having a very good collaboration, all the people with my lab and all the people in the clinician. So thank you so much.
Thank you very much, Dr. Andreani, who guided us to the complex world of matching or mismatching in hematopoietic stem cell transplantation. Usually there are no questions for the invited speakers, but if there is one very hot question, we could accept it. Γιούλα. Γιούλα, λέω, μήπως θες. Γιατί έκανε παράπονα. Το μικρόφωνο. Εντάξει.
I want to congratulate you.
Thank you.
Because all the time you were talking, I had the feeling that somebody is telling us who has the knowledge, but who is using it in favor of the patients. And this is very important. A typical question would be, I know that registries are keeping their files during the years. Are there any diagrams to show what the survival of the patients is during the years? I mean, from the 80s, 90s, 2000 and so on, to have the overall survival, and then look at the reasons that they form it. Thank you.
Yeah, it's a very important question. Thank you for the congratulations. It's really nice to share with you and all with Greece friends that I see in the audience, these things that we debate every day when we meet in our conferences. Yes, of course, there are, and I didn't take care of this, also because we do not have our own registry in Rome. We just take care of the work of, we accept the work of the others, but we have a very nice relationship with the Italian registry, of course. And I have listened many times, Nicoletta Sacchi, that she's the leader of this group, saying that in the last few years, the clinical outcome of a patient has improved so much respect to the 80s or into the 90s, because the typing and the drugs and the experience that all the clinical centers have developed are very useful. So if this was your question, the answer is yes.
Okay.
If you want, I'm here. I do not have any problem in.
Absolutely, an excellent talk, an excellent speech, an excellent presentation. I don't know what to say. Included everything on
Microphone.
so an excellent presentation. Thank you, Dr. Andriani. It included everything about hematopoietic stem cell transplantation. Okay. And really, it makes things complicated and complicated and complicated.
It's true.
Nothing is easy anymore in this field. And I wonder if there is a, I would say, point system indicated what to take into consideration in, in several cases, because we know that different case is to transplant a young patient comparing to an older one, a male, when comparing to a female, one disease and another disease, acute myeloid to chronic myeloid leukemia, whatever. Is there, say, something to show us the way, or to show to the clinicians the way, how to manipulate each case?
Yeah, there are very clear indication about these. Yeah, in the future for sure. But for the issues that you were mentioning, like age, of course, the better, the younger the donor is, the better he is. This, of course, the sex match is very important, as well. Also, the consideration of the CMV from the donor and for the recipient. This is for sure a very clear indication. What is not clear any longer is the compatibility level of HLA, because the presence of the donor-specific antibody, the DSA, the possibility to transplant patients that are different from not only from one locus, but from a full haplotype. This is really incredible. If you turn your head to the 10 years ago, you would have been killed in proposal, something like that. So the introduction of post-transplant cyclophosphamide has really done a great job. Still, we consider, I consider that the work of Esteban, the work of EBMT, in which even if you improve the outcome with using the post-transplant cyclophosphamide, you still can improve it much more if you use a 10 out of 10 donor. This is one of the things that is still debating. And it's very important to understand, because the post-transplant cyclophosphamide is using now not only in the haploidentical setting, but also in the other. And then this is what we need to further investigate, and then to enlarge the option of what is called the permissive. That's why I wanted to bring you with me in the story of a permissive mismatching, because that is very important. The story started with the DPB1, but everyone didn't consider, if you remember, at the very beginning, we didn't even type the DPB1, and instead it has become important. And on the basis of the model of the DPB1 has been developed what is called the PBM in the class 1. And this is again, very something that we need to work for the next year all together with these Greek friends that I have here. So I'm not sure that I will pronounce it Eferisto. So thank you really very much.
Thank you again for accepting our invitation to come.